Welcome to the latest JETZON open call. As ever, we have two excellent talks. The first of those is from Nestor Arandia, and he's presenting some work also done with Niculina Musat, who's also on the call. She can answer questions on the more technical matters later. But otherwise, Nestor, over to you. Please tell us about those CNN flux from the aggregates to the bacteria. Thank you.
Yes, thank you. So I'm going to share my screen. Okay. Now, well, thank you very much for inviting me to give this talk. I'm very excited. My name is Nestor Arandia Niculina, and currently I'm a postdoc at the Marine Science Institute in Barcelona. But here today, I'm going to present a work that I started back during my PhD studies, during a short stay that I did at the Helmholtz Center for Environmental Research in Leipzig, Germany, with Niculina Musat, and also continued working on during my first postdoc at the University of Gothenburg with Helmholtz. So this particular work, it's a result of many collaborations with different people from different institutes. And I'm very glad to say that it finally was published this September in Scientific Reports. So in this paper, our aim is to analyze the efficiency in the nutrient transfer from marine diatom aggregates to their colonizing bacteria in order to understand how the succession of different phylogenetic groups of colonizing bacteria may impact the carbon and nitrogen cycling within these diatom aggregates. So first, I know that a lot of people are here in the audience have advanced knowledge about marine particles and also the microbial activity, but I would like to start this presentation giving a very brief introduction. So as we know, phytoplankton, and particularly diatoms, because I'm going to focus in diatoms in this talk, are key players transforming the inorganic carbon and nitrogen into their organic forms and putting this organic carbon and nitrogen available for the environmental bacteria. Besides, phytoplankton can also aggregate between each other, mostly thanks to the production of transparent exochromic particles, forming large aggregates that will eventually sink, transporting part of this organic matter to the twilight zone and below. Bacteria are also extremely important players in the evolution of these aggregates. For example, first, the bacteria can facilitate the formation of these aggregates by promoting the production of the transparent exochromic particles, but also bacteria are responsible of the breakdown and the degradation of these particles by their exoenzymatic activity. And of course, liberating a big amount of dissolved organic matter to the surrounding water. However, there are still a few things that we don't fully understand. For example, how different colonization dynamics work for different phylogenetic groups of bacteria, and most importantly, how the succession of these colonization strategies affect to the carbon and nitrogen fluxes within the aggregates, and also to the carbon and nitrogen, organic carbon and nitrogen would be exported outside of the aggregates. So to answer these questions, we planned some experiments that were conducted mostly by Isabel Plawon. First, we prepared axenic cultures of the diatom here, Leptocylindrus danicus, which were grown using exclusively carbon-13 labeled bicarbonate as carbon source and nitrogen-15 labeled nitrate as the nitrogen source. Once the cultures were ready, during an oceanography cruise here in Cape Blanc in the coast of Mauritania, we also collected environmental samples of marine bacteria, which were co-incubated together with the diatoms into roller tanks. So the function of these roller tanks is basically to facilitate or promote the aggregation of the diatoms. So here in these tanks now we have carbon-13, nitrogen-15 pre-labeled diatom cells together with environmental samples of bacteria. We keep these incubations for 72 hours, and we took samples every few hours to measure one, microbial abundance, to follow how colonization of different bacterial groups was evolving, and two, to measure the carbon and nitrogen transfer from the diatom cells to the colonizing bacteria. So as I said, first we measure the microbial abundance of different phylogenetic groups of bacteria using cardfish. In this case, we targeted 4 different groups. First, alpha proteobacteria, Bacteroidetes, and Alteromonas and Pseudoalteromonas, as I showed here. So in these 2 figures, you can see how the bacteria are within these diatom aggregates very clearly. So once we measured the abundance of both of the different phylogenetic groups and also located the exact positions of each individual cell, we started analyzing the carbon and nitrogen transfer from the pre-labeled diatom aggregates to the bacteria at single cell level using a secondary ion mass spectrometer known as the nanoscales. So thanks to the nanoscales, what we can do is we can have let's say like isotope, we can have the isotopic composition of each individual cell, thanks to their like a huge spatial resolution. Another advantage of the nanoscales is that we can measure up to 7 different ions simultaneously. For example, in this image, we can see the carbon-12, carbon-13, nitrogen-14, and nitrogen-15 ions. So using a combination of these ions, we can also, for example, measure the carbon-13, carbon-12 ratio and the nitrogen-15, nitrogen-14 ratio. Using these ratios, we can quantify the amount of carbon-13 that was, or also nitrogen-15 that was incorporated by each individual cell. So here now, I'm showing some of our nanoscales analysis. In the first image, I show the sulfur-32 isotope, which we use as a proxy to estimate our microbial biomass. So in this image, we can clearly see the diatom cells, the Leptocylindrus danicus diatom cells, and their associated bacteria. And now, using the nitrogen-15, nitrogen-14, and also the carbon-13, carbon-12 ratios, we can also measure first the amount of carbon-13 labeled bicarbonate and nitrogen-15 nitrate and carbon-13 bicarbonate that was incorporated by diatoms, and also the amount of diatom-derived nitrogen-15 and diatom-derived carbon-13 that was incorporated by each individual bacteria. Here, for example, we can see that these bacteria were like a highly enriched in nitrogen-15, and although here it's not very easy to visualize, we also observe like the carbon-13 incorporation by bacteria. So, but while we're analyzing these samples, these microscopy samples, but also the nanoscale samples, we realize that not all the bacteria within the aggregates were following the same lifestyle. So because of that, we decided to divide our bacteria into 3 different categories or 3 different lifestyles. For example, here first, we have bacteria that were found clearly living within the aggregates, but were also physically attached to the diatom cells, meaning that bacteria will like a direct physical interaction with the diatoms. We call this fraction the attached bacteria fraction. Second, we also have bacteria that were, again, clearly living within the aggregates, but in this case, they were not physically attached. But instead, they were like embedded into some kind of organic matrix, most likely transparent exoplanar particles, as you can see here in this helium ion microscopic image surrounding the diatom cells. We always found some kind of organic matter. So in this case, the bacteria that were not directly attached, but embedded in this organic matrix, we call this fraction the non-attached bacteria fraction. And finally, we also found that some cells were outside of the aggregates, as you can see in this image. We found that some cells were like living, let's say, far away from the diatom cells, and we call this fraction the free living bacteria. So this last fraction, the free living ones, we don't consider these bacteria for our analysis. We only focus in the attached bacteria and the non-attached bacteria fractions. So now I'm going to start showing our results. First, I will focus in the bacterial abundance. As I said, we measure the bacterial abundance for different groups: Alteromonas, Pseudoalteromonas, Bacteroidetes, and alpha Proteobacteria, for both the non-attached and the attached bacteria fractions. So, and in this first fraction, the non-attached one, first, we observed that at the beginning incubation, some alpha Proteobacteria were growing, but these alpha Proteobacteria were rapidly replaced by rapidly growing pseudoalteromonas cells. And they became immediately after 30 hours, they became the dominant group in this non-attached fraction. For the attached bacteria fraction, we also observed something very similar. But in this case, pseudoalteromona was the dominant group since the beginning of the incubation. So these results indicate that pseudoalteromonas was actually the primary colonizer of the diatom aggregates, which may also indicate that pseudoalteromonas may have like an important role in the first stage, like early stages of the aggregate degradation of the aggregate breakdown. However, after 39 hours, we also found increasing abundance of Alteromonas, and they became the most abundant group by the end of the experiment. So here we are seeing that in the second stage of colonization, Alteromonas were able to rapidly colonize bacteria. So we define these Alteromonas as a secondary colonizers. So then we started analyzing the carbon and nitrogen transfer from the diatom cells to the bacteria. For that, we decided to exclusively focus on the most abundant phylogenetic groups, which were Alteromonas and Pseudoalteromonas. So in these two figures, what we are showing is the nitrogen-15 enrichment rates and also the carbon-13 enrichment rates for the three groups: diatoms, Alteromonas in green, and Pseudoalteromonas here in blue for the different incubation times in the A, A and C panels. But also we are showing here in the B and D panels the average enrichment rates for the different incubation times. So the first thing that we see that is that, as expected, the diatom cells show with the highest nitrogen-15 and carbon-13 enrichment rates. However, also for carbon-13, we barely found any difference in the Alteromonas and Pseudoalteromonas carbon-13 incorporation. For nitrogen-15, we found that during the initial stage of the colonization, Alteromonas group was highly more enriched in nitrogen-15 than Pseudoalteromonas. Actually, Alteromonas was nitrogen-15 enrichment in Alteromonas almost was as high as the one found for diatoms. So that's a big enrichment ratio. This indicates that during the first hours, the first 48 hours of the aggregate colonization, Alteromonas was the primary nitrogen consumer. However, this is contradicting because we just observed that Pseudoalteromonas was the primary colonizer of the aggregates, right? So, but still, we would expect that nitrogen-15 enrichment rate be higher for Pseudoalteromonas. So in this case, we're seeing that it is actually higher in Alteromonas. So we think that this mismatch is related to their colonization strategies. For example, as Pseudoalteromonas is the primary colonizer, they had to incorporate like high amount of nitrogen, for example, to boost their colonization abilities by increasing their abundance, but also maybe to synthesize enzymes to degrade the diatom aggregates. For that, they have to incorporate also not only the diatom-derived nitrogen, which is what we are seeing here, but also non-labeled nitrogen from the seawater, which means that they are incorporating also other sources of nitrogen. On the other hand, Alteromonas as secondary colonizers, they basically they were taking advantage of this primary colonization and aggregate breakdown that pseudo-Alteromonas was doing, relying basically exclusively in the diatom-derived nitrogen. So here we are seeing that the presence of different phylogenetic groups does actually affect a lot to the nitrogen sinking within the aggregate, not so much the carbon cycling. But we think that besides the phylogenetic groups, the different colonization strategy can also have like an important impact in this nutrient cycling. So in this result, we are showing the combination of both attached and non-attached bacterial fractions. However, we were also very interested to see if there was also some kind of difference in the nitrogen and carbon incorporation between both fractions. So here, we are showing the diatom-derived nitrogen incorporation rates against diatom-derived carbon incorporation rate for Alteromonas here in green, pseudo-Alteromonas in blue for the non-attached fraction shown as filled dots, and the attached fractions shown here as the empty dots. So the different dots indicates different time frames. So here, while almost no differences were found for nitrogen incorporation, at least no significant differences, for carbon incorporation, these differences were clear with the attached fraction incorporating between 2 up to 3 times more carbon than the non-attached fraction. So this indicates that this attachment strategy, even within the same aggregate, the ability to be physically attached to the diatoms could be like a very advantageous in terms of carbon incorporation. This is very interesting because in a work that we published back in 2017, we saw something very similar. We also calculated the nitrogen-15 incorporation, the phytoplankton-derived nitrogen-15 incorporation against phytoplankton-derived carbon-13 incorporation rates, in this case for the free-living bacteria. These are not within an aggregate, they are free-living in the seawater, against bacteria that were physically attached to the phytoplankton cells. We saw the same result. No effect for nitrogen, but in the carbon incorporation rates, we found 3 times higher incorporation rates in bacteria that were physically attached to phytoplankton cells. So again, these both results highly indicate that this attachment strategy is like a very convenient for bacteria in terms of carbon cycling. So as a conclusion of this presentation, we have the 3 main highlights, right? 3 main outcomes. So first, we found that pseudo-Alteromonas may be one of the first responsible bacteria in terms of diatom aggregate colonization, which implies that they may also have like an important role in the aggregate breakdown. Besides, we found that the nitrogen assimilation within the aggregates, diatom-derived nitrogen assimilation, depends on the phylogenetic group, but it also depends on the colonization strategies, with significant difference in isotope uptakes between the early and secondary colonizers. And finally, the ability to physically attach to the phytoplankton cells can also largely determine the efficiency of the alkyl-derived carbon uptake, with up to 3 times more carbon incorporation rate observed in the attached bacteria in comparison to the non-attached ones. So this was our talk, and here I would like to thank first the Helmholtz Center for Environmental Research and the Provis Lab, and of course Niculina Musat, that they basically funded this big part of this, the nanosims analysis. University of Gothenburg and Helleplog, which because also I think that they had the original idea. They started this project, and also of course Dr. Hugo Berthelot, who was also co-first author of this paper, and he did an amazing job with different calculations. Yeah, that's it.
Thank you very much. That's extraordinary. Actually, have the technology to do that. So thank you very much for that talk. I see we already have one question. Cordelia, do you want to open your mic up and just ask it directly?
Can you hear me?
Yes, we can.
Perfect. That was a really great talk. Thank you. And it's great to see techniques like this being used within a lab setting and also with environmental relevance. I just wondered if you accounted for the bottle effect in terms of the bacterial abundance and then the potential dominance of certain taxa as a result of that.
Sorry, I didn't answer very well. The dominant groups in our experiments?
Yeah, so with the bottle effect that could potentially happen by using closed systems, did you account for that in any way, or?
Well, yeah, that's really hard to measure in this case because, well, at the beginning of the experiment we didn't have like a lot of bacteria, but here our incubation lasted only 72 hours. So then well, I don't think that's like a long time to measure a bottle effect here, but I mean, definitely may happen some bottle effect, but if we are, yeah, that's really hard to measure in this case because we are basically focusing on bacteria that was specifically attached within the aggregate, and we observed that the same groups were present since the beginning. So these bacteria, we observed that they were growing in these aggregates. So there was not a big change in the community since the beginning. So, I mean, that's of course something that always can happen, but still we think that 72 hours and also most of our results that we gather happened in the first 48 hours. So we don't think that it's a long period to observe like an important bottle effect.
Okay, super, thank you.
Thank you. Any other questions? I can't see any in the chat at the moment, but does anybody else would like to ask anything? So I've got one, which is, it's probably sort of wishful thinking, but one of the interesting hypotheses around the processing of particulate carbon, and I thought that Tom Anderson might be in the call, so he may want to comment on this as well, is the idea that zooplankton may be feeding off the bacteria on aggregates. So this is related to the microbial gardening idea. So I'm just wondering, I mean, does your technique allow you to measure the efficiency with which the bacteria are taking up carbon from the diatoms? I'm just wondering about what fraction of that carbon goes into new bacterial cells and what fraction of that might go into, for example, dissolved organic carbon.
Mm-hmm. Yes, so that's one of the calculations that Hugo did. So here in this graph, what we're showing is the percentage of diatom-derived nitrogen and carbon. So here what we do is considering our carbon-13 and nitrogen-15 that was in the diatoms, which was very stable, as you can see, like that was pretty stable. We can more or less know, according to the signal enrichment, carbon-13 enrichment and nitrogen-15 enrichment that we found in the bacteria, we can measure the percentage of carbon-13 that was incorporated by bacteria that was coming directly from the diatoms. So there was that was kind of more or less simple calculation because we already know how much carbon-13 has the diatoms. We measure how much carbon-13 it's being incorporated by bacteria, and in this case, the only source of carbon-13 was from the diatoms because the bacteria were not incubated with the carbon-13 bicarbonate and nitrogen-15 nitrate. So in this case, we can measure how much of that, in terms of percentage in this case, how much of that carbon ended in the bacteria. That's part of the calculations that we are showing here.
Okay, so
maybe to add a few things, that it will be very difficult to analyze what you just asked. Okay, so you need to really separate the fractions. So we have this analysis at single cell level, but you have to consider that it's a pretty dynamic process. So you would need, first of all, stable, you need to choose your isotopes wisely, and then to separate the cell fractions, and then to have a very well-designed time series in order to see how much of the carbon ended up in bacteria, and from that bacteria, how much ended up in the zooplankton.
Yes, to know like exact amount concentration of carbon once incorporated, yeah, Niculina is right. There's a lot of things that we need to know. Also, concentrations of other organic carbon that are in the environment that the bacteria may be using, not only the algae with carbon, nitrogen.
Yeah, I thought it's a simple question for something that's difficult to achieve. I thought that would be the case, but yeah, I mean, nevertheless, it's a very useful insight in terms of the potential efficiency of the process. You know, I mean, particularly, you know, the graph that you've got on the screen there, the difference between the non-attached and the attached in terms of the fraction taken up is quite striking. Are there any more questions or comments before we move on? So just another comment from Cordelia that, yes, sort of referring to the microbial gardening hypothesis. Right, so thank you very much to Nestor and to Niculina once more for presenting that.
Thank you too.
As if any of you have any questions or comments, I'm sure that both of them would be delighted to, you know, receive them. So feel free to email them. And as I say, if you want to have another look at the talk later, we'll hopefully post it on the YouTube channel tomorrow or the day after, I would hope. But thank you, Nestor and Niculina.